We'll start off, honored to start with Yossi. Take it away.
If I speak, there is some microphone somewhere here. There. Okay, thanks for the invitation. It's really great being here for the first time. So this talk is going to be very unusual. I think I've never shown so many new slides in a talk in my life. So before I start the actual new talk, I just want to say a few words about our work in general, because I'm not going to talk about this too much today. So I like to say that we study decision making. I'll do this very briefly. This is a kind of a framework that I like to think of. So take the model organism, your favorite model organism. Humans could be also one. And the question is, how do they make decisions when they have so many channels of information coming in, whether it's sensory, individual sensory, social information, cognitive capacities, environment. And we mostly think about foraging decision-making, just because it's something that is rather easy for us to quantify. So, where do I feed? Should I go to where I ate yesterday? Should I visit several? If it's a fruit bat, should I visit several trees? Should I visit one tree tonight? Should I fly with other individuals? These are kind of decisions that the animal has to make again and again, night after night. And we try to study this. I'll explain in a second how. Why bats? So of course, there are many interesting animals. I think that if you want to study a mammal, then bats are, I would say, advantageous because of their diversity, actually. Of course, marine mammals are also, they are also important and interesting, but much more difficult to study, I would say, even though bats also pose some really serious challenges. They're tiny. Most of them weigh less than 30 grams. And again, they fly, they have remarkable longevity, a remarkable immune system. But for me, it's the diversity. So there are 1,500 species of bats, and you could probably say that they're the most diverse group of mammals. I hope I'm not offending any rodent scholar in the room. And each of these bats is a strategy, okay? It's a sensory system, it's a story. And if I'm interested in sociality, Michael mentioned sociality, navigation, foraging, there are interesting species to study and available also. And now the approach that we take usually is instead of bringing the bats into the lab, which is something we do as well, but trying to take the lab and put it on the bat. So we have these tiny sensors that we use, and they're actually multi-sensors, so they include usually some kind of a tracking system, whether it's GPS or something else. We can, let's see, there it is. So we can record, for example, physiology. This is body temperature. By the way, look, this is interesting. A bat elevates, body temperature goes up by several degrees within minutes every time a bat takes off. So very unique physiology. This is acceleration down here, physiological measurements such as heart rate. And we can even quantify the environment. So in this case, we're measuring ambient temperature by putting a sensor on the bat. And actually, you can see nicely, you don't know this area, but we are actually managing to quantify urban heat islands, okay, using bats as sensors. So I promised David to speak about echolocation, okay? So echolocation 101, you all know this, but just let's go through it very quickly. Most bats echolocate. I will talk about bats that don't echolocate today. And what they do is they emit sound, right? But interestingly, sound returns, so sound is reflected from everything, right? And the returning signals might be very complex, like what you see here. So there are two ears, right, two microphones, and each of these peaks can reflect or might be an echo returning from anything in this environment, okay? So this is a superposition of many, many, many reflections. And the bat's task is basically to identify, if it's an insectivorous bat, to identify within this cacophony of echoes, right, the right echo, and also to localize it within this highly cluttered environment. And this is something that no human made machine can do today, okay? Take the most sophisticated, military machines that exist. They cannot get even near this performance of a bat, and for sure not with the size of a bat, okay? So, the equivalent human machine would be, 100 kilos, and it will not perform as well. Just, if you want to listen to this, maybe for the first time in your life, so this is the echo. Make it a little bit louder. That was the echo of an apple tree. I'll explain later how we record it. It's, of course, downsampled because it's all ultrasonic, so you can hear it. But I just wanted you to get a sensation of the complexity, multiple reflectors. And here's a cornfield. You can actually hear the rows of corn, okay, the multiple reflections. Okay, so I recorded these during my PhD, and I'll get back to this later on. By the way, if there's any important question, just ask. Now, when we put the microphone on the bat, we usually, okay, so our sensor has a microphone. I didn't say that's maybe the most important sensor for us. And when we put it on the bat, we can infer a lot of the bat's behavior. So these are all spectrograms. I guess you're familiar with them. So here we can see these are echolocation signals. Okay, they're around 26 kilohertz, right? So we cannot hear them. And here are echolocation signals of what I would call the focal bat, the bat carrying the microphone. And I can also see the conspecific. So I can look at interactions with other bats. And this is something I will talk about in a second. Okay, I can sometimes also detect the prey. In this case, it's a frog-eating bat in Panama. And these are recordings of the prey, of the frog itself. That was very short. Maybe I'll try again. Very short and very faint, but it sounds like something like that. It's a tundra frog. The bats love them. There's a very interesting story there of sexual selection, right? So I don't have time to go into it, but the frog, so the signals that the frog emits, right? The frog is trying to get a female mate. The signals that it emits that the female prefers are also the ones that the bats are best in detecting. So there's a whole trade-off there, very interesting, but no time for it now. These down here these are attack sequences. I'll show you more in a second. So this means that the bat is attacking. You see that it's increasing repetition rate. It's emitting more calls per second. And also the signals are changing. Okay? And finally, I also know sometimes if this is a successful or not attack because these are chewing sounds right after the attack. Right? So I can infer all of this for an animal flying, 1,000 meters above sea, which is something very difficult to do when we study animals in the wild. Here's the whole picture. So what you will hear now is the attack. This is we call this it's a molossus bat, and you will basically hear this sequence again downsampled. So you will hear the calls becoming more frequent. You'll hear a lot of wind. You can also say something about the maneuvering based on the sounds of the wind, and try to figure out yourself whether this was a successful or an unsuccessful attack. I see some people got it already. So it was successful, right? You can hear the chewing. By the way, you can hear chewing, echolocating at the end, chewing, echolocating. You have to see while you're chewing. Maybe I'll play it again for those of you who are not that fast in echolocation. Now, it's a beetle, probably a big beetle. If you still cannot imagine what it looks like, this is probably what it looks like. It's not the same species, I must say, although there are no experts here, but still. Yeah, so, yeah. Okay, so the first thing I'm going to talk about.
Can I ask
yeah, sure.
Anything about the prey type based on the attack sequence?
No. The attack sequence, no. It's a good question because it will be related to information. So we don't see. I would say roughly the attack sequence doesn't tell us much about what is being attacked, and I think it's because it evolved, during 50 million years to sense, and it's good in everything more or less. And it's, that was like as a PhD, I was always looking for these very subtle changes, and I don't think they're there. But based on the chewing, we can say quite a lot, yeah. And people are starting to do that now, yeah. Okay, so one of the most difficult situations for a bat is the following. Okay, so many bats are social. They roost in these colonies. These are greater mouse-tail bats. You see they have a tail, which is rare for modern bats. We think this is similar to the ancestral bat. This was a falcon. And they're it's sunset, and they're just about to go out, to fly out. These are, they're like 5,000 females in this cave. what you heard, by the way, are social calls. We'll go back to that later, not echolocation. That's the Lake of Galilee down there, right? You can see it in the horizon. So something like 2,000 years ago, right? Jesus was walking on the lake, right? And then he saw the bats coming up, and he asked himself two questions, right? So the first one is how do they avoid interference or jamming? And I'm actually going to address both of these questions today. So after many years of wondering how do bats, okay, so I didn't say this, well, it's actually the next slide. So these are bats coming out of that cave, actually a nearby cave, but the same species and the same kind of behavior. And after many, many years, we managed to track dozens of bats, which I think this is probably the largest number of animals tracked within a collective, I think, at least using GPS or something like that, not using video. People have done this with video, I should say. And we also have microphones on part of the bats. This is great work by Aya Goldstein, today already Dr. Aya Goldstein. And if you put a microphone on the bat, this is what you get. So it's a complete cacophony, okay? Try to get, you're looking for echoes in this. These are echolocation calls of many, many, many individuals around you. And you're supposed to detect echoes within this cacophony, okay? So this is what it sounds like, again, downsampled. And many people compare this to, by the way, on the right here, you can see some kind of a distribution of the spatial distribution of the swarm. You can see already, and this is important, that they rapidly spread, right? And then the density goes down, right? So within 100 meters from the cave, or a little bit more, they already spread. And many people compare this problem to what we call the human cocktail party problem, which is the following. Right, so you're in some cocktail party. I recorded this in a conference, actually. And you're trying to follow one individual, maybe a few individuals. In this case, the bats also have to localize them because they want to avoid collisions, right? And I should say, people, and if you watch the movie, people always know that the bats hardly collide in these very dense situations. So what we've done is to take the acoustics and to then develop a sensory motor agent-based model. I'm not going to go into all of the details because I have quite a few other topics that I want to talk about. And if you're interested, we can discuss the details. But basically, what we see, so this is a recording, I think, 100 meters from the cave, where it doesn't look like this anymore. It opens up a little bit. And we can quantify, so these are focal calls. This is the bat carrying the microphone. And you can see different types of interference. So there's what we call just general masking, when another bat is calling within the sensory channel. And there's jamming, and that's when another bat is calling, another bat is calling when you are expecting to receive an echo. Okay, so we know exactly the window. So the bats are not foraging, they're just flying out, I assume, at this point of time. And again, by the way, they can use vision, but we're asking whether they can solve this problem using echolocation. And we know exactly when echoes from other bats will arrive, and we know how to model that. And we can ask when is this blocked by a call of another bat. By the way, I should say the echo, of course, is much, much fainter than the call of a neighboring bat. That's the big problem, right? And we can model all of this with a very simple. So the approach our approach is always to use an underestimate of everything, right? In terms of the capacities of the bat, and an overestimate of all of the problem as much as we can. And then if the model can solve it, then we assume that the bat can solve it as well. And to make a long story short, here's an example of the agent-based model. And indeed, we show that bats can fly in these environments, I would say even easily, avoiding collisions. Now, this is the focal bat. You can see down here, this is the acoustic information or the acoustic scene from the point of view of the focal bat. Okay, so echoes in green. Red means masking. Black means jamming. So every time it is jammed, and you see it happens a lot that it is jammed, this means that it completely misses the information about an echo from a nearby bat, okay? So still, how does it, you see there's a lot of jamming. And masking means basically that there is some kind of interference and the information will be, so your estimate will deteriorate. You can't estimate position of other bats as well as you should. So if you ask me to summarize still despite this, how do the bats manage in this environment? So I would say maybe there are 3 points. One I already mentioned, they spread out quickly, okay? And very quickly this acoustic scene becomes this, okay? It becomes an easier problem. Second, you have high frequencies, focal hearing, 2 ears, okay, which narrow the whole problem. You can see it here. You can't see it because of the projector. I see it. But there's a gray, wow, you really can't see it. There's a gray triangle around the focal bat showing the area, the relevant volume, where the bat is actually receiving echoes from other bats. And it's much smaller than the large problem. It's not equivalent of this. And the third thing is a lot of redundancy. So these bats call this environment, they will call 50 times per second. Okay, you asked me about the rate of the calling. So it does change all the time, but not depending on the type of prey, but more depending on the density and the clutter. And that's really, really, I would say, hardwired and very. By the way, we even have. I'm drifting here, but we even have an experiment where we show that the bat will collide with a foam wall. So there's a big foam wall, and the bat will just fly into it, and it will do this again and again and again, several times, okay? And we show that the echolocation rate does increase, okay? So there's some kind of below perception, if you want, area in the brain that does receive echoes and respond to them, even though for the bat there is nothing in front of it. So that's why I say. And there are also experiments from the 60s where people dissected parts of the brain, and they showed that this is still intact, this increase in repetition rate. But sorry, that was a side story. So back to here, there's a lot of redundancy. So even if 1 echo is blocked, or 2 echoes are blocked, or 50% of the echoes are blocked, as we show here at the beginning, okay, around here, they can still, our very simple sensory motor model can solve this. Okay, so that was just a little bit about how bats, if and how they can solve this very, very challenging task. And now, to connect this to the topic of, I think, of this meeting, I said, okay, let's talk about echolocation and its potential for communication, right? So we talked about sensing so far. And the truth is that we know a lot. We know that echolocation can be used for communication, okay? So we know that bats eavesdrop on other bats. And we have several studies that have shown this, and others have shown this as well. And we know that bats can probably fly in groups. They benefit from flying in groups, especially when they are searching for food that is ephemeral, that is hard to find. And they can do this using echolocation only. You will not hear a single social call, okay? They will fly over many, many kilometers using only echolocation as a mechanism to fly in a group, okay? So they're using it also for communication. They also know when somebody else is attacking prey based on those sequences that I showed you, and they will use that too in order to find the prey and try to intercept it. We model this again. I'm sure many of you are familiar. Sometimes it's called the 3 radii model. So it's again a simple agent-based model where the agents behave differently with other agents depending on the radius. So either they avoid the collision if it's very close, or they align with if it's in a second radius, or they just are attracted if it's a third radius. And there are different versions of this model with and without alignment. And if you run a model like this, you get this kind of behavior. So these are bats exiting. These are bats we studied. This is supposed to mimic bats that live on islands and forage in the ocean, in the Sea of Cortez. But it could also reflect any other species. But that's why they're all leaving the island. And there's food somewhere in the ocean, fish. Flocks of fish, okay? And it's very hard to find. You have a huge area. You cannot predict where food is. It's a very difficult foraging or detection or finding or search problem. And if you just use these very simple rules, right? All you do is pay attention to nearby agents, okay? You get this type of behavior. So you get these groups. And you can see that the first group now found the food, okay? And the other group now finds the food, and this bat now knows nothing about food. It's only following the individuals around it, and you can see that most of the individuals then will find the food just because somebody around it found the food. So this is, in one slide, this shows us how echolocation can and is used by bats constantly for communication. So this is a searching swarm, if you want. right, without any direct communication. So nobody is shouting, there is food, nobody is shouting, come here, nobody is shouting, I'm Yossi, follow me. None of this, only echolocation. Okay, and they do it very, very...
This is your agent.
Yeah.
This is not the real bat.
Yeah.
It's your agent that you are conjecturing is, if it can do it, then the bats might be doing the same thing.
Right, so I didn't put it here. I have a very cool movie showing this for the real bats in Isla Partida, Sea of Cortez. Unfortunately, it's not 50 bats flying together because we always have the problem there are 10,000 bats leaving the island. We put 100 GPS devices, we get 50 back, and the chances of getting 2 together are, you know, you would calculate it better than me. They're very, very low. So we always have to infer, right? I gave the reference to the papers. Yeah, we do the thing I should have, there's one movie that I should have shown and which would really clarify this. We use the microphone a lot. So even if that's one of the big advantages of the microphone. So even if I didn't tag all bats, I can hear when there are other bats nearby. Right? Thanks to the microphone, you could see it actually up here. Maybe I'll just go back to that for a second. And that allows us here. You see, so here, this is the bat carrying the microphone. This is a conspecific, anonymous conspecific. I don't know who this is, but I know when I have conspecifics nearby, and then I can do, and that's what we did, a lot of statistical modeling and see whether any null model that does not assume intentional aggregation can produce such a density, and the answer is no, not that I have been able to find. So yeah, it's a good question. I thought you're all computer scientists, so I'm only showing you the computer stuff. But yeah, yeah.
Do you think that they're using other bats, echolocations for their own navigation? So
yeah. So that
neighboring bat vocalizes, but I'm hearing the echo and I'm using that as well.
Yeah, that's one of the big questions in the field. It's called bistatic radar. Okay, in radar this is being used since World War II. The thing here is that signals, because it's ultrasound and it attenuates very fast in air, it's all very short ranged. We're talking about meters. You'll see this in a second. We have not been able, we tried and others have tried. I've never been able to show this that this happens in a lab experiment, for example.
But you think it's happening?
I don't think so, no. Oh. I think it's too complicated to compute. I did think, so one situation in which I thought it might happen is when you have many bats, and maybe there it does happen, by the way, and maybe there's an obstacle, a big obstacle, and you hear something, and it gives you some first indication. So something very, very rough. Maybe that's happening, and that we haven't maybe tested. But the cool idea that you hear the echo, and then I think probably not.
The argument against this is because it's mono-channel, right? Because it's sound. Because we do that all the time with vision. So I can use other waves that are reflected by somebody else's flashlight.
Yeah, if we were using flashlights.
So taking it back out of the channel is impossible. It's harder.
It's not mono. It's bi. You have two. Yeah, but exactly. You have to compute where this is coming from, and you don't know where the source is. Would be very difficult. Yeah.
Did you use the model of the previous one, 2 back?
Yeah.
You can mask out. So if you artificially reduce from 50 times a second to whatever, 20, and then allow them to see the mask noise.
Allow them to see the mask?
Well, I'm not sure exactly how the model works. I'm sorry, I haven't read the paper. But so you take out their echolocation, but give them the information that's contained in the noise that's masked.
Yeah, I don't know if we did that exactly, but thanks a lot for the question, because I was supposed to say the reason why I like these agent-based models is that, assuming that we're doing something that is similar to reality in some way, I can do exactly these types of experiments, which I can never do in reality. So what we have done, for example, is to ask, okay, what if I completely remove the effect of masking? And so now bats, let's say they don't hear the calls of others, but they do need to avoid collisions with them. And then I can ask what is more important. Is it the avoidance of collisions? Because there are other things moving there, or is it the acoustic interference? It maybe sounds like a detail to you, but these are arguments that in the field we had for you. We still have, even, you know it is. Even if we publish something, other people still keep their opinions. So we've done a lot of those. I think we actually did look at bistatic, but not exactly like you suggested, which would be interesting. Yeah.
Cool. Yeah, thank you.
Okay, so I showed you that echolocation can contribute to communication. What about the other way around? Can they use social calls? I showed you there are a lot of social calls for echolocation, for sensing, right? So this is a project we started a year and a half ago with Miriam Knonchin. I think she also maybe spoke in one of the CETIs, but she's doing a lot of work on bat communication, babbling. If you read about babbling in bats, that's her work. And the way we do is, or one of the ways is, I already hinted to this before, we can put a speaker, right? That's how I recorded those tree echoes. I can put a speaker and that emits, and in this case, I'm even using an ear, a 3D printed ear of a bat, and I can put them and microphones in the ear, you see, and I can put different types of texture or reflectors. So there's a wall, there's a vegetation, and there's a moth, which is a very small reflector, in front of it, and I can rotate everything and just look at the echoes. And now I'm instead of broadcasting echolocation signals, I'm broadcasting communication signals, like, for example, human speech, right? And again, and this is work in progress. We're just analyzing this. But these are social calls. They might look a little bit similar to echolocation calls, probably not coincidentally. I'll say something about this in a second. But what you see here are social calls of, 5 species of bats, and the echoes, okay? So the rectangles show the echoes. So clearly, we get echoes. I think this is for the wall, right? There were 3 different types of reflectors. The wall, obviously, is the most reflective, but only from 90 degrees, right? By the way, in vegetation, that's a big difference. Vegetation will give you a lot of echoes from everywhere. That's a huge difference. And you see that we clearly get echoes for all emitted social calls. So you can get echoes when using social calls. And this, of course, makes us think about the evolution of echolocation, because obviously social communication was there before. We don't know what was the precursor call of a precursor bat 80 million years ago. I don't know. If you have any ideas how to find out, that would be helpful. So we can only look at modern calls. And you see that clearly they reflect echoes. By the way, all of these are much lower in frequency than the echolocation calls. If you're asking what's the big difference between this and the echolocation, usually the frequencies are lower. They're also longer calls. So duration is important and frequency. And another thing that we can do and have done is to look at the ontogeny, right? That's also a kind of a window into evolution sometimes. Evolutionary processes, and we took the ontogeny, we recorded pups for one species from day 0. You see up there, day 5, day 7. These are social calls. By the way, here you can see nicely how these social calls, they start at 20 kilohertz, and over time, the fundamental frequency goes up to almost 40 kilohertz. So that's the main polishing, or I don't know what's a good term to describe this. What's happening to the development, right, that we see of the calls. And all of these will produce echoes. I'm not going into all of the details because I see I'm running out of time. But all of these would produce clear echoes. Yeah. Now, yeah.
How do you know that echolocation predates social calls?
No, other way around. So.
Oh, sorry.
I don't know for sure, right? But I do know that first echolocating bats appear in the fossil record 55 million years ago. The estimation is that they started 60 million years ago. By the way, there's a lot of debate about flight and echolocation, which one preceded which one, without any agreement, or maybe together. And we know that all mammals have, or nearly all mammals have social calls, and they go much further back. So, I mean, it's very likely, right? But I don't know, of course. Now, as I said, one thing that is very important is duration. So frequency is important. Frequency allows you to see small moths. That's why echolocation uses high frequencies. But duration is also very important because, when I'm talking right now, I'm receiving echoes. I can put a microphone in my ear and we'll all see this very clearly. But if I receive the echoes while I'm talking, this complicates their processing very much, right? Our brain, of course, ignores echoes. That's one of the things it does, right? Unless they return in very, very late. But so duration is important, and these signals had to become shorter. And I think, and again, this is still things we're thinking about. They're new. But I think that most of the social calls that we see today that bats use have already went through some process of becoming shorter because they might be important for sensing, as I'll show you in a second. And here's one graph that suggests this. So these are we just took a lot of social calls from the literature of different groups, okay? And you can see that's duration, and it's just, box plots, distributions. And you see that bats tend to have very short calls in general. By the way, these are shrews, and some people also think that they might be using echolocation. There's a few papers about that, even though it's still under debate. And here's an example to kind of even strengthen this idea. This is a molossid bat in flight. These are echolocation calls, and suddenly it emits a social call. This is a social call, okay? Because there's another bat nearby. Okay, I know that because this is the other bat. And you see that this suddenly creates a gap in sensing, right? There's a gap. This is the next echolocation. This is the previous echolocation call, and there's this gap in the middle. And I just proved to you that this is still useful for getting some echoes. Okay, so it might be that the shortening of these social calls is driven by the need to when you're flying and emitting social calls, you might still need some sensory information, and you might be able to get it from using these social calls that look like echolocation calls, basically. Okay, but as I said, this is all still kind of thinking about the problem. Now, if I do have a few more minutes, I want to show you something a little bit different and to take you to a few thoughts that I've been having about animal communication in general in the past few years. So this was all relevant. Everything I talked about so far was relevant to echolocating bats. But as I said, there are like 150 to 200 species of fruit bats that do not echolocate. Some of them have some interesting form of echolocation. These are Rosetus bats, which Michael also studies here in Berkeley, and we also work on them in Tel Aviv, and they actually use tongue clicks. So there are a few theories whether their ancestors lost echolocation and regained it or not. Not so important for today, but they use, this is their form of echolocation. They have some adaptation to emit these in much higher frequencies than I can, even though this also creates a lot of ultrasound, but theirs are even better than mine. And as you can see, they are part of the phylogeny. They're not a completely separated group, but you can see where they are on the phylogeny. And their social vocalizations look very differently from the rest of the bats. They look like this. They're much, much, much noisier, okay? And in a second, you'll hear a lot of them because we worked on it quite a lot. So they're noisy vocalizations. I mean, much less tonal, okay, even though they do have a clear fundamental. And here, actually, you can see an echo. So this is I'm not even sure that we recorded the echoes like we did before for these vocalizations. Somewhere here, the echo begins. Maybe it's this thing here, but it's very difficult to process because they are longer, as I said. And we worked quite a bit about on the communication of these bats. If you go into a colony with thousands of bats, this is what it sounds like. Okay, so they're constantly chattering. And very, very quickly, we've done some work on the ontogeny of these vocalizations. The pups start emitting what we call isolation or distress calls from day 0. And over a period of almost 2/3 of a year, they move from these vocalizations to the adult vocalizations, and they emit these sequences. You see here a sequence of vocalizations. And if you take any acoustic feature, we've tried many, and here we're plotting the adult. This is the repertoire of the adults, and then we can look at the full ontogeny of the pups within this feature space. And by the way, this is all data available, so I think this is quite unique. We have the entire repertoire of the pups from day 0. This was recorded in the lab because we wanted very, very high quality recordings, and until almost a full year. So this, and we have processed it, but not enough. So I think this is a very unique data set. And we've also looked at these vocalizations using machine learning, so Gaussian mixture models in this case. And we only looked at snippets of the calls. And we've shown that potentially there's a lot of information, okay? Statistically, there's information, so we can identify the emitter of the call, and we can identify the context of the call. So what do I mean by context? So we had students who really became experts on these bats, watching, hundreds of thousands of hours, and identifying 4 main types of interactions, a few others, but those are the main ones that we've seen. So this could be either, I should say most of these vocalizations, you already saw it in the colony, are what I would call, what I think bioacousticians would call agonistic, right, or aggressive. So the bats are roosting, I say it's like if you grow up in a football stadium, probably most of your communication would be about move out of my way or something like that, right? So I mean, some people are disappointed that these bats are not, serenading to each other, but that's that. So here are the contexts that we identify, and so there's feeding. So they're fighting over a piece of banana. Okay? And there's a mating. So this is the female, and the male is trying to mate, and listen to her response. I don't know why it's so weak. Maybe somebody can turn on the volume, but you get it. This is just fighting over space. And finally, during sleep, sometimes you wake up your neighbor, and that's his response. Yeah, that's my favorite. And they all sound the same to us, right? And now, as I said, we applied this statistical learner, right? And we managed to, far above chance, right, identify context, okay? But what does this mean, right? That's my question. And now I'm going to share a few thoughts. And I'll be glad to hear the opinions of the machine learners, human machine learners in this room. So my argument is that we can play with the statistics a lot and find differences, and we have very powerful tools to do this today. But we need to develop ways, and I'm a biologist, right? I need to do this to show that these classes or statistical differences are really important for the animal.
Yes, we turned the volume up, so maybe you can play this again.
You want to hear the waking up again. So here's the mating. Here's the banana. Yeah. Thanks. And go back to sleep. Yeah, so as I said, we need to find ways to show that this is relevant for the animal. And measuring the response, so the best thing I think would be to measure some kind of behavioral response, to play back these different classes and show that the animal responds differently. But why should it, right? Basically, it's listening to an argument between two other bats, right? So why should I respond differently, right? So we do see a response. Bats will twitch their ears, but it's very hard to measure a differential response. By the way, we even generated down here, we generated using a gun model. This was years ago, before we had the years ago, right? Probably 3 years ago, before we had LLMs and stuff like that. So it's but we managed to generate an artificial sequence, and the bats do twitch their ears to this one too. This is what it sounds like. Yeah, it's not bad. I can even tell the difference, but it's not bad. But again, my point is that we need to find ways to show that the bats care about these differences, right? So here's one way, theoretically, that we could do it. This is what we call a habituation task. So we habituate the animal on one signal, and then we switch, and we want to see a response, right? So and you look at the pupil. It will dilate when we change the signal. Okay, it works quite nicely. I don't know if you can see the pupil. Now it will open. You see? But this was so it definitely works, right? It's an autonomous response. But yeah, when I play different contexts, I see no difference. Okay, so I don't manage to measure a response to these different clusters that I found statistically. It doesn't mean that the bat is not perceiving them differently, right? I cannot measure it. It's very difficult for me to measure it. And my argument is that that we need to find ways to do that. Because if we don't identify the units of the communication, right, we cannot go to the next steps, right? So here's something that many people do. I cluster communication signals somehow, unsupervisedly, for example, and then I plot some very cool statistics, right? And then sorry, I don't want to offend anyone. I do this too, right? But what does this mean? So what I'm trying to say is that what we really need is so this is the way I picture it. Okay, we have some communication system on the left. We have access to this feature space. It can be acoustic, it can be visual, if it's visual communication or factory communication, whatever. And in feature space, and we detect clusters somehow, right? So either we do it based on the acoustics themselves, right, with some kind of a learner, or we use context like I try to do. But still, this is context detected by human eyes, right? But in the real perceptual space, these signals might be clustered completely differently. Okay? Suddenly, maybe they reveal some reference or context. Maybe the order suddenly becomes important, maybe not. So if I want to ask questions about syntax or regularity, right, or concatenating, I need to know the units. And I don't think we currently know how to do that. And if you're still not convinced and you're thinking, yeah, but acoustic feature space must tell us something, right? So things that are similar in acoustic space, they must be similar perceptually. I'll try to convince you using a very classical animal model that this might not be the case. Okay, so this is Jonathan, is my son. He's nearly 2 years old. He's trilingual, so he's growing up in a French, we're in Montreal at the moment, French, English, Hebrew environment. But like my wife says, he doesn't speak 3 languages. This is what he's non-fluent in 3 languages. This is what he sounds like.
אנחנו פשוט לא מבינים אף מילה שאתה אומר. באיזה שפה זה?
So he's clearly trying to communicate.
באיזה שפה אתה
I'll
מדבר?
translate my wife is saying we don't understand the word, and he's responding. And I
אף
think,
מילה אני לא מבינה.
as an animal behaviorist, I think that he thinks that he's conveying some messages.
עכשיו הבנתי.
אמא, את לא...
It's complete
עכשיו
gibberish,
הבנתי
okay?
הכל.
There's nothing there that you can understand,
באמת הבנתי.
except for that. That was Maya, his sister, okay? So there you could identify something. So he's somewhere in between complete gibberish and a few word-like vocalizations. And now I'm going back to this. So there's an urge to convey a message, right? And we have access. So in this case, we also have access to this space, more or less, right? Because we share a perceptual space. And let's play a game. So these are a few vocalizations from a sequence that Jonathan uttered during the night with some specific context that I think I know. And let me ask you, which one of these would you, and there are more. I just gave a few examples. There are like 10 of these. Which ones of these would you cluster together, right? I don't know. I actually don't even know. But if I run it through an algorithm, it would cluster them, right? For sure. And I would argue that all of them actually conceptually or semantically, they're all the same. Okay, here I'll try to convince you. I think your perceptual space is similar to mine. And if you listen to them, you'll see that they're all exactly the same. Okay, so I think, I don't know if I hope you agree, they're all exactly the same, although statistically we would probably cluster them into different clusters. You think this is only a case of maybe, prelingual, sorry. This is like my nightmare at night. I don't want to listen to this again. You think this is only a case of prelingual vocalizations? So now here's Ima, which is mother. Okay, so a little bit more similar, I would say, but still, if you cluster this, you would probably get many different clusters, although it's all one very specific vocalization. In this case, it's actually a word even. Okay, so as I said, I think my point is that it's very difficult to detect the units today, and using algorithms that run in feature space, the available feature space might not be the best way to do this. I also wrote here, with animals, we also have, so with animals it's completely impossible because I have no access to their perceptual space, unless maybe I use physiology or something like that. I'll talk about it in a second. We have much more variability with animals. We have dialects, right, which makes everything even more difficult. And there's also, I think, a lot of context ambiguity, right? So eventually we want to have a language, right, to decode their language. So we also want to know what they're saying. But this sequence, this I, one day it could mean one thing, tomorrow it could mean a different thing, right? Again, I like to think about Jonathan, about toddlers, because I think it becomes, it's much more intuitive for us. And I know he uses the same sequence for many different contexts, so there's a lot of ambiguity. And moreover, tomorrow he might ask for the same thing with a different sequence, right? So there's no one-to-one mapping at all, okay? So linguists always tell me, by the way, I apologize if I used horrible terminology with linguists in the room, but people always say with humans, we also don't have one-to-one mapping, this word can mean that and that and that, of course, but it's not even close, I think, to this ambiguity that we see here, I think. I have to listen to this again. Okay, so to summarize, and then if I have 2 more minutes, I'll show you something else that has nothing to do with all of this. I'll just skip the summary, I think. So I showed you the dual use of echolocation and communication signals. I talked about the challenges of using machine learning AI to decode animal communication. I think we need to think of how to get access to that perceptual space. Physiology would be one way. Of course, if we could use behavior, that would be great. Physiology would be one way. Physiology is not a miracle solution. Even if we record the entire brain, we don't know that this is a one-to-one representation of the perception. I actually argued this before. So it doesn't solve the problem. We are trying to use MRI. You can see two bats. Unfortunately, I don't have yet functional results. This is just anatomy, but these are two bats lying like this, one next to the other in an MRI machine. It's a top view. You can see their eyes. And if I have one last minute, I'm going to leave the animal kingdom, or not completely leave it, and talk about plant signals for a second, because I think we're involved in very interesting work that has recently come out, and maybe a few of you might be interested. So a few years ago, my colleague Lila Hadani and myself discovered that plants are constantly emitting ultrasonic, I should not say signals, right? Because they're not signals in the evolutionary term. They're just acoustic signals. So actually, it was known, if you look at the literature, forever, more or less, that plants do things that are called, so they don't do it, right? There's cavitation in plant water vessels. That's one phenomena that creates vibration. People have placed piezoelectric pressure sensors or whatever on the plant and have recorded vibrations, but for some reason, nobody tested, as much as I know, if it also, if the energy goes out into air and if the signals are airborne. And that's what we did, okay? So, I mean, it's not some miracle. It was known. Just nobody put a microphone, an ultrasonic microphone outside the plant, and that's what we've done. And we found that plants are constantly generating these sounds. Around, as you can see here, around 50, 60 kilohertz. We also show that these sounds are, they convey, potentially, they have information about the status of the plant. So a drying, dehydrated plant will emit, I don't know if it's really generate many more of these. The rate goes up dramatically, which makes sense if it's cavitation, by the way. We don't know for sure if it's cavitation. We can also detect differences between plants, which also makes sense if the vessels, have different diameters, for example. So there's potentially a lot of information. I'm not saying any of this is intentional, even though Lilach is testing this, right? But, and it could be really interesting to think of how a plant would use this and evolve to use this, but there's no evidence. But just recently, two of my students, Guy and Raya, bless you, we've decided, okay, so once this is out there, we decided, okay, there are animals that can hear these signals. That was, for me, the main incentive. Moth can hear these signals. Bats can hear these signals. Many rodents can hear these signals, and others. And we had to start somewhere. We decided to start with moth, because moths use plants to lay eggs. Right? They lay their eggs on plants, and it's super important for them whether the plant is dehydrated or not. And I'm not going into all of the experiments. We have a lot of controls for this. I'm going to give you just the main one. What we do in this experiment, we have a moth in this tunnel. You see it's 1.5 meters long. We have signals, drought, tomato signals played on one side. We have no signal on the other side, and we count the number of clusters and number of eggs that the moth lays along this axis. And you can see down here very clearly, and there's a feeder in the center, at the center. So you see that the moth, and this is results for, I don't remember, 50 or 100 moths or something like that. You see that the eggs are clearly, she has a very clear preference, and this repeats. We've done this 3 times, 6 months apart with different setups. And there's a clear response to the sound. Okay, so either she'll either lay the eggs near the feeder or on the side that is playing sound, which, by the way, is a bit confusing because the sound suggests that the plant is dehydrating. So you have to read the paper. We also have some, we refer to this confusion also in the paper. But, and we also have some other sounds, control sounds, which she does not respond to. Okay? So this might be, I'm very careful because we need much more validation, but might be a first indication to an acoustic interaction between, or an acoustic response of an animal to a sound emitted by a plant. We know that they respond to volatiles, right? We know that there's chemical communication going on, and of course visual communication going on, but nothing about acoustic communication between plants and animals. And I'm not saying this is communication, right? We still don't know that it's...
What's your explanation why it goes
Yeah, good question. You know, this was very frustrating, but we repeated again and again. So what we did is we repeated this experiment, but now with two dehydrated plants, okay? And one of them has a playback. And now the moths prefer the silent plant, okay? So that makes sense. If you have two plants, they're identical. The problem is we can't control olfaction, right? So that's why that's why I wanted to go only with the acoustics as a sensory ecologist. I said, no, let's isolate the stimulus, but then we got this strange result. So we placed, so when we place two plants that are, as much as we can say, identical, and one of them is emitting sounds that are associated with dehydration, then the moth will prefer the other side, okay? That's very clear. Why is there a reversal of the behavior? The hypothesis that Raya, my postdoc, suggested, that's the best one that I have so far, but I don't know, I'm not saying, you know, I can't prove it, although we did do one control to try to prove it, is that maybe in the absence of any other cue, so there's no plant in the room, nothing, maybe those sounds are interpreted by the moth as the presence of a plant. So the moth knows that the plant emits these sounds, and she's searching for a plant, and if there's nothing else, and the control that we did is we used a very dehydrated plant versus nothing, and we showed that they do prefer a dehydrated plant when there's nothing, when there's no alternative. But that's just an interpretation.
You tried maybe changing the amplitude of the signal?
We play with the rate.
So you have like two, and one is stronger?
No, no.
One is maybe more hydrated, so that it's not just like, I don't know how do you...
It would be the rate, I think, and not the amplitude that would change.
The rate is higher
Yeah.
versus
The thing is, this is a very difficult acoustic environment already to begin with. It's small, you know, to get these signals on one side and on the other is already challenging. I think that would be even more difficult. I finished. I have to thank students, a lot of students, great students who've done a lot of this. You've seen some of their names. Shameless promotion, my book about bats, if you want to read more, is coming out in October. And that's it. Thank you very much. Do we have time for questions? Or?
Well, thank you, Yossi. I just we did get some questions in there, and we want to stay on. Any burning questions?
Yes.
Yes.
Oh, just a couple comments. So your example of the fruit bats in the dolphin porpoise world, which are different taxonomically and a bit socially, they're, you know, the dolphins are making whistles and click pulses, et cetera. The porpoise, no whistles, right? Just clicks. Mm-hmm. So clearly, many animals are encoding information in these creative ways, but relative to their environment. Mm-hmm. Second comment, when you're talking about looking for the smallest unit of sound to then look at structures and sequence, whatever, we've been playing with that over the years. And a linguist once looked at our stuff and said, you know, because of the variation you showed with your son and the clumping, et cetera, it's like maybe take a top-down approach instead of looking for something like phonemes or something that adds up to, you know, words or sequences. Take a top-down approach because we can't even do it with humans, shall we say, right? So I think that's an interesting thing to think about when we're looking at sounds and breaking it down. Maybe a bigger picture, too, is really going to be important to look at, which you're suggesting, I think, anyway.
Mm-hmm. Yeah, I agree with those comments. I mean, yeah.
Thank you. I guess let's go to Adam.